Hey, welcome back to the channel. This is Joel Duff, and you've stumbled upon an episode of Critiquing Creationism. Actually, this is where we look at something that creationists have said and try to learn something from that, right? Try to be a little educational here. We're not just here to mock or make fun of, what creationists are proposing here, although there's certainly room for that, especially in this one today. But mostly I want to draw out something about what is actually kind of a difficult concept, I think, for a lot of people to understand. That is how we compare genomes, one genome to another. How do we assess the similarity or dissimilarity between genomes? And that is something that I've just been recently lecturing on in my genetics course. We've been talking about genomes, genome structure, and so forth. And so I've been emphasizing this point that I'm going to make in this particular video. But let's start out with what triggered me to maybe say, hey, it's time for me to talk about this topic yet one more time on this channel about genome similarity. And that is, I've seen this image along with the others I'm going to show you in a moment coming by my social media stream again. This is Creation Today. This is Eric Hoven's Creation Ministry. And I've seen this particular image multiple times in the last week. And I just look at it and go like, what exactly does this mean? And does this data make any sense at all? Why does it make sense even to Eric Hoven? I get what his point is here, or at least what he's trying to say and communicate, but his data on this slide makes no sense at all. Let's just think about what is being, without reading his words first, right? You share as much DNA with a cat, 90% of your DNA, 70% with a slug, 50% with a banana, 98% with a mouse. Well, why the 98%? Where does that come from? Why 98% with a mouse when only 80% with a cow, or 90% with a cat and 60% with a chicken? Where do these numbers come from? If you did a search for percent similarity, or I just said a word that I shouldn't have said, percent has a meaning that we're going to look at in a moment. I guess the first question of it, what is this saying? Yeah, that's saying percent. Presumably it's percent similarity of DNA, but how is that calculated? And where in the literature would you find 98% similarity between a mouse and a human? Actually, I think you can find that number. And you could probably find most of these numbers, but here's the kicker. Each of these numbers comes from a different form of calculation, a different method of calculating the similarity of a genome to another. And so these are not apples to apples comparisons. They're not even apples to bananas comparisons. They're not humans to bananas comparisons. They are not the same calculation. Therefore, these percents can't be compared to one another until you know what they are. So that's what I'm here today for, is to try to explain to you in hopefully fairly basic terms how different methods that geneticists, genomicists used to try to evaluate the differences between genomes, or how similar genomes are. Now, what Eric Hoven here is doing, he's saying humans share DNA with cats, cows, and even bananas. True. All right. That's a true statement. We do share DNA, nucleic acids. And then he says, if similarity were to equal ancestry, which of course he doesn't believe there's ancestry between all these different organisms, but if similarity were equal to ancestry, then, well, obviously we'd be half banana, right? Because we share 50% of our DNA with bananas. Again, we have to talk about what that 50% means. Absurd, of course, right? So similarity points to design, not random descent. But where does that 98% for a mouse show? Does that mean we're not actually very dissimilar from a mouse? Haven't you heard that we're 98.5 or 98% similar to chimpanzees? And, we have pretty similar morphological appearance to chimpanzees. I'm afraid I don't really feel like I look or seem like I'm like a mouse, as similar to a mouse as I am to a chimpanzee. So it just intuitively, even these numbers don't really make a whole lot of sense. And so I just want to remind you that I have talked about this, and we've talked about genome similarity mostly in the context of humans and chimpanzees, and how young Earth creationists have been beating this drum about the dissimilarity of chimpanzees and humans, and have calculated in a variety of different ways. And so I've that's 29 minutes, 28 minutes, an hour and I've done a couple other videos on that. Gutsick Gibbon, a YouTuber, has also spent multiple hours talking about how similarity is determined between humans and chimpanzees. But what we're going to talk about here is just a multitude of different organisms, and generically, how does one go about determining the similarity between two different genomes? And why those responses? Because, again, Creation Today, I think this was on the very same day this was also posted. Are humans really 98% chimp? Or is it just that evolutionary hype dressed up as science? The lie, 98% similar. Again, what is 98% similar? How is that calculated? That's what we want to talk about here. And okay, keep this in mind. There's 98%. And then how about this little ditty from Creation Today? I think on the very same day as those other ones, posted the DNA gap gets wider. The differences in man and apes are 48 million nucleotides. Sounds really impressive when you say it that way. 48 million nucleotides. Wait a second, 48 million. Have you divided the size of the human genome by 48 million and gotten a percent? Hey, I have. I punched it into my little calculator right here. That's a pretty simple calculation. And you know what I got? 1.45%. 1.45%. Wait a second. So the similarity between a chimpanzee and a human based on this image right here, this number that's being presented by Creation Today is 1.455%, which is, I'm sorry, isn't that about 98.5%? Wait a second, let's go back. Are humans really 98% chimp? The 98% lie? But apparently they're that different, which is 98.5% similar. This is a head scratcher for me. Why this is, what the message is here. And then he tosses in this little thing here, just 3 changes in nucleotides causes death. So if we're 48 million differences, then it's been many deaths over again if we had shared a common ancestor. I guess that's the message. This is young Earth evidence. Yeah, I mean, yes, a single nucleotide can change death. I could point to tens of thousands of individual nucleotides in your genome that if I were to change that one base, you wouldn't be alive. But the 48 million base pairs that are different between humans and chimpanzees most of those are not very significant differences. They make no difference, really. It comes down to just a few differences, in fact. Oh, well, see, there again, I've already slipped into talking about how do you compare genomes? Do you compare genomes as if every single base pair were equal to each other and say that if you have X number of differences, that's significant? What makes significance? Or do you have to assess each individual base or individual portion of the genome and ask how important is this to the function of the organism? And then maybe only compare the functionally important regions of genomes and say, are there differences in those regions versus maybe a stretch of DNA that makes no difference at all? It's relatively neutral in the genome and just kind of there. And if it doesn't differ between 2 different genomes, or if it does differ between 2 different genomes, you're kind of like, do I really care about the similarity? Do I really care about the difference? Probably not. All right, so let's say that we want to sit down and we want to just ask ourselves, how similar are 2 different species? And let's not do humans and chimps. Let's just say wolves and coyotes. All right, you've got wolf and coyote. Those are different species. They each have a genome. And you'd say, like, how similar are those genomes? How are you going to go about doing that? You might say, Joel, hey, just sequence the whole genome. Find all the base pairs of a coyote, find all the base pairs in a wolf, line them up together, look to see where they have differences in A, T, C's, and G's, add up the differences, divide by the total length of the entire genome of those 2 organisms, and boom, you got a percent. And that would tell you that they're maybe 1% difference or 0.9% difference. I don't know what the difference is. For one thing, that might actually not be calculable the way I just said, right? Because there's a little problem here. Their genomes aren't exactly the same length, right? If they're not the same length, then that means one of them has extra base pairs compared to the other. Or if you want to look at the other direction, one is missing base pairs that the other one has. And so let's say, just for ease of calculation, I'm just doing this on the fly here. Let's say that the wolf is 1 billion base pairs in size, and the coyote is 1.1 billion base pairs in total length. If you were to just add up the total number of base pairs in its genome across all of its chromosomes. When you get done with that, you'd say, like, well, one of them has 100 million base pairs more than the other. 100 million base pairs more than the other. So if you just divided the size of one genome by the other, you'd be right at, well, close to 10% difference already. You'd say like 10% of the genome of this one organism doesn't even exist in the other one. So therefore, you could count those all as differences and say, like, they're all different. And that's not even without looking at the lined up sequence of that they actually do share and calculating the number of individual base pairs that are different. So that's going to add to it. You know, you're probably going to end up with 10, 11, 12% differences between a coyote and a wolf. And yet we just saw Creation Today saying that you and I were 98% similar to a mouse. So how could a coyote and a wolf be 11 or 12% different? Now, let's say so there's an alignment problem where they don't have the same size genome because some of them have extra base pairs than others. So you have a question that you immediately have to answer. How are you going to account for that difference? Well, it depends on what the nature of those differences are potentially. What if the difference is that just one segment of the wolf genome is duplicated? So 100 million base pairs are copied over. So you have 2 copies of that 100 million base pairs. And you might look at that, that's just like one real difference, right? You just copied one whole segment one time. That's one change in history between the two. We don't want to go down the rabbit trail of, like, does having 2 copies of that much DNA make any difference in development and so forth? It probably does. But if you have 100 extra million base pairs and they're just a copy of something else, how significant is that? How should I count that as a difference between the 2 organisms? And really, in between any 2 organisms, you compare things and you're going to find out that there's gaps in one, like an area where you don't have something aligned. Between you and me, there probably are tens of millions of differences in terms of just the length of our genomes. So you and I are several percent different from each other. If you just do a really crude analysis to determine percent similarity, if it's just based on overall total number of shared bases. All right, so you can see that it's not trivial to do this because each genome comparison you do is going to have different challenges. Now, what you see in the background behind me is just a quick slide that's thrown from my genomics class from a while back. And so I don't want to go into this much detail, but I'll just give you the basic categories. You could try to do a complete alignment-based sequence, right? And that's where, and you can do that for, say, like our mitochondrial genome. You know, you have a mitochondrial genome, I have one, 16,000 base pairs. 16,400 and something base pairs or whatever. Yours might have 1 or 2 extra bases, or I might have 2 or 3 extra bases that you don't have. So that's a fairly insignificant number of, like, extra bases, like that don't fit, that we can't compare, that we can't align with each other. However, for the most part, we can line the whole thing up, in which case we could then figure out in that alignment, which hopefully is like, you know, you got this particular gene, it's 700 base pairs long, and you have 700 base pairs, I have 700 base pairs. We think that's the same base pairs in their alignment. They're homologous with each other, which would be the word we'd use. And then you would just go along and say, like, well, how many of those 700 different? He was like, you got 2 differences compared to me. And then we add up across the entire 16,000 base pairs, and you and I might differ by at most 100 differences. But probably we only differ by 15, 20, 25 differences in our entire 16,000 base pairs. And you get a percent for that. We could talk about the percent similarity of our mitochondrial genomes between each other. And that would be a relatively simple, straightforward way of comparing all of us. And I could compare my sequence with chimpanzee, which is about 1,400 differences. So it's a lot more differences than between you and I, the interspecies variation difference. Okay, I'm talking too many other examples. Let's get back to the basic message here. So the point is, you could try just lining up the whole genome, but that's rather difficult to do between any two organisms that are moderately different from one another. They're probably going to have significant differences in their total sizes of their genome. They might have different numbers of genes. They're going to have rearrangements such that some of them have genes that are flipped around in the other direction. They're going to have duplications of portions of their genomes. So they have two copies of the same gene. So you'll be like, well, I can line up these copies with my copies. But then what do I do with these other copies, even though they might have almost the same DNA sequence? Do I count all, 5,000 base pairs of that copied region that has the same sequence as this other copy? Do I count that as 5,000 differences, or is it one difference because it's one duplication event? You have to decide the ground rules, right? When you begin to formulate, like, how am I going to present my percent similarity, right? How am I going to present how similar these two organisms are? And you need to state what your methods are. And this is one of the big problems with most creationist calculations. And they're throwing around these numbers of percent similarities, is they rarely tell you, like, where did that percent come from? Sometimes you're just like, I got that from the literature, but they don't understand what the method was that was used to do that. Oh, that's exactly what happened here. The 98% similarity between a mouse and a human being is based on lining up all the common genes, right? The genes that we have in common, a lot of core genes that all mammals share. And so a bunch of homologous genes, you say, like, they got an insulin gene, I have an insulin gene. You've got an olfactory receptor gene, whatever, and I have an olfactory receptor gene, whatever, right? You have an ATP synthase gene, I have an ATP synthase gene. You find those genetic elements in the genome, line those up so you can think of it as the coding sequence, the genes, the genes that make proteins. And you line those up and you ask, how many similarities are there? All right. And how similar are they? Well, they're about 98% the same for the core genes that we share. Now, of course, mice have some genes that I don't have. And you and I have some genes that mice don't have. That, in this case, those aren't included in the calculation of how similar we are in our shared sequences. So in other words, we share a sequence, but that sequence isn't identical. We're 98% similar in our shared sequence. We've only covered one method, and I think I've shown you it's a little difficult to do. I mean, you kind of have to make decisions, right? And if you make a decision, then that is an assumption you're making, or it's part of your process, your methodology. You put it in your paper, you talk about, here's what I did. And then what you're hoping to do is you're hoping to say, like, I'm going to compare a coyote and a wolf. And what I'm going to do is I'm going to look at all the protein coding sequences. I'm going to look at all the genes in both of those. And I'm going to compare the sequences of those genes. And if I'm going to compare all the sequences of the genes that they have in common, I'll give a percent for how similar those genes are. And then if I'm going to compare all the genes and include genes that one has that the other one doesn't have, or copies of genes the other one doesn't have a copy of, I'm going to show you how I'm going to calculate that. I'm going to get a different percentage for that. Right. Then I could also do this sort of ortholog gene-centric perspective. Actually, sorry, that's actually what I've been talking about. The alignment-based sequence is like, I'm aligning everything. All right. And so I'm aligning all the regions, even between the genes. I'm aligning as much as I possibly can. I'll take chromosome number one from a wolf and a coyote. I'm just going to lay them across each other. And what I'm going to find out is that I have a whole bunch of genes, and a lot of them are in common, and they're kind of in the same places. And I just try to match up as much as possible. But there's going to be all kinds of disjointed regions where I have extra base pairs, I have missing base pairs, I've got, and so forth, right? And so you're just going to calculate the similarity of all the sequence that you can align and hope for the best. And you're going to get, honestly, not a terribly useful answer out of that for reasons I'll show in a moment. And then I've been talking about the ortholog sort of gene-centric point of view. And that is like, let's look at shared identity among shared orthologs, the same genes. And between a coyote and a wolf, they likely share 99.9% of their genes, right? the things that make proteins, and they probably also share 99.9% of the regions around genes that are the regulatory regions. They're going to have different variants in those. That's the spice of life. Different variants in genes and different variants in regulatory elements. The way we turn off and on genes, or how much we turn up the volume and down the volume of how we use our genes. That's really what makes us different from each other, less so than like I have actually completely different genes that do different functions, at least between closely related organisms. And so we're gonna take all those regions and we're gonna say, like, this is the important stuff of an organism, right? This is the thing that makes an organism have the characteristics that it has. All the regulatory regions and the protein coding sequences and the non-coding RNA regions that are coded for by DNA. And we're going to take all those that we know of and we're going to compare the sequences of those and get a sequence similarity. Or we might just say, like, how many in total of all those items do they have in common? We don't care about the sequences. We just want to know, like, out of all their microRNAs, they might have several hundred in the genome. How many do a wolf and a fox, or a wolf and a coyote share? All right. So again, you have to explain the ground rules. What is the thing I'm actually comparing? And then there's other methods that are like statistical methods. There's an older method in which you do hybridization. You take the two genomes and you basically melt them, right? And you see, well, you can compare melting temperature. You know what melting is? I know it's a weird sounding thing, but there is such a thing as a melting temperature for DNA. And that's essentially like, as you warm up DNA, right? You're going to reach a point where the DNA starts to separate into two strands. So you go from double-stranded to single-stranded. And that's going to be determined on the number of ATCs and Gs, right? The percentage of ATCs and Gs. If you have more Gs and Cs in a genome, they have 3 hydrogen bonds holding them together. I mean, they're weak bonds, but nonetheless, 3 is better than 2, right? A's and T's, all right, have 2 hydrogen bonds, which they're trying to like hold each other in the double helix. And so the zipper starts to break as you heat it up, right? You add energy and eventually the hydrogen bonds break. And so TA bonds are going to break before CG bonds break or separate. And so if your genome, right, has more C's and G's in it, it's going to have a higher melting temperature. And another genome that has fewer C's and G's will have a lower melting temperature. And there can be significant differences between the composition of a genome just at the level of how many A, T, C's, and G's are there. But that's usually not between species. Species usually have very similar ATCG content, like you and I have very similar ATCG content. And the difference between species, even other primates, is probably not enough to really see a significant difference in melting temperature. But if you're comparing, dogs and sheep, it could be an actual difference between the composition of their genomes at the level of base composition, and therefore the melting temperatures will be different. So that's a whole different way of getting, like, you get like a number based on melting temperature, which reflects a difference in genome composition. You can also look at amino acid sequences. If you want to look at an even more conserved way to ask, like, what's functionally different between an organism in terms of their proteins, you could look at amino acid sequences. And you say, well, yes, the DNA can differ, but it still might make the same amino acids, in which case, your gene is not different than mine. I'm sure there are places where you have a DNA sequence that's different than my DNA sequence, but it still might make exactly the same protein, which has exactly the same function, in which case, do I really care about those differences at the DNA level? If we're making functionally the same thing? No, you're not going to care about that, right? And so you might say that the really important differences between organisms are at that amino acid level. Now, I would come back and I would tell, my genetics students would say, Dr. Duff, yes, we knew about those protein things we talk about, but then you've been beating it into us that it's, your genome isn't just your protein coding genes. It's also the non-coding RNAs, which are often regulatory elements, which tell genes to turn on and off. And that's the thing that often determines, like, the different tissue expressions and different, that's why, muscle cells different than a neuron is often in those other regions. You have the same DNA, it's how you're using it. All right, so there again, you could have two organisms that have incredibly similar proteins, and yet they might be utilized in different ways in different species because of the way that they're being regulated and turned off and on, and expressed would be the word we'd want to use here. And so the last one over here, this little green box right here, marker locus-based method, is one that I've often mentioned on this channel because it's a convenient sort of way of looking at it, taking a quick look at how similar two things there are to each other. And the locus might be like, in this case, a locus could be the whole mitochondrial genome, but that's small compared to the nuclear genome. But you could take just one gene. You just ask, like, if I sequenced this gene that like all mammals have, cytochrome C or insulin gene or, ATP synthase or something like that, right? Some housekeeping gene. And I compared that between you and me and a mouse and a sheep and so forth, right? And a cow, like I'm trying to think of the things were on that picture before. And I compare the sequences of those, right? Just that one gene. That one gene might be, and this is the caveat here, is it might be reflective of sort of like the overall similarity between our whole genome. You don't want to be really careful doing that because some genes are more conserved than others and will accumulate differences much more rapidly than other genes will. But nonetheless, you could just do those types of comparisons as well. And so you can find all these different kinds of comparisons. I haven't even mentioned all the ways you can do this, but you can find lots and lots of different ways that organisms have been compared at many different levels of their genome. And there is no universal consensus of like, here's the method. When somebody says, this species is 67% similar to this other one at the DNA level, kind of saying at the DNA level tells you at least they're talking about DNA and not amino acids. But how did they calculate that? Right? It's really, they got to tell you. They have to tell you. And then when you hear another percent for in another paper where they're doing some kind of comparison, you have to ask yourself, are they using exactly the same method? And this has been the big criticism of Tompkins at Institute for Creation Research. And how other creationists have used that number. They said, look, he found it was 15% different. Human and chimpanzees are 15% different. Whereas those other biologists say it's only 2% different. See, they've been lying to you. But it's an apples to oranges comparison. The method, the number you hear that's 98.5% or 98% or 98.8%, that method is this ortholog gene-centric method. Saying we're lining up all the common genes, which are most of the genes, and asking what are the differences in those protein coding sequences, right? The primary parts of your genome that constitute what makes our physical being the way it is. And we're going to look at that portion of the genome and compare it. And that's where we get that 98.5%. And again, if I did that with you and me, I would get 99.9 something percent, right? You and I share all the same genes. We share all the same important components of our genome. On the other hand, if I did the first method and just lined up the sequences and asked if I just line everything up and I look at all the gaps and extra bases you have and inversions you have compared to me, and you've got like 10 million base pairs over here that I'm missing. And then you started saying like, well, what's the percent similarity overall in total of the genome? You and I might only be 94% similar, 93% similar, probably 95% similar. Right? But that doesn't sound nearly as interesting, does it? When you start saying we're that different from each other. But honestly, we're not that different from each other. All that extra stuff that makes us look that different is insignificant material. It doesn't constitute anything really significant about how you and I differ from each other. The differences you and I have from one another, certainly we have significant morphological appearance differences, right? And clearly other, metabolic differences and so forth. But they come down to individual, usually fairly small changes. By small, I just mean single nucleotides. A single nucleotide can make a big difference in the function of a gene, but it comes down to just some changes in our genes, not changes in our like massively repetitive sequences that are the same sequence over and over and over and over again. And you have 20,000 copies of this repeat, and I've got 18,524 copies of that particular repeat. And so you and I differ there. I don't really care. All right. It's not making any difference in terms of who I am as being different than you. Okay. I probably should have brought this up earlier. Why do I save the good stuff for the end of the video after all of you have tuned out? So I wanted a quick example of how crazy things can get and how creationists using simplistic methods can run afoul very quickly of reality, right? So the reality here is, here's two species of salamander. Okay. These are two closely related salamanders. The redback salamander lives from, in my area, up into Canada, sort of like East Coast, sort of northerly. And Plethodon idahoensis is what it sounds like. It was discovered in Idaho, and it's found in British Columbia and Montana and a few other locations north of the border. They do look pretty similar, don't they? I mean, these are pretty similar looking species. I suspect that given these two found in the same area, which they don't overlap, but if you did find them in the same area, if you didn't know salamanders, you'd just think they were just variants of each other. But they're good, solid species in that they're different enough that they don't interbreed. They don't produce successful offspring. Now look at the genome size, 67 billion base pairs. Let that sink in, 67 billion base pairs in one copy of their genome. You have 3.3 billion base pairs in one copy of your genome. 67 billion. That's almost 30 times the size of your genome. And by this is not the largest genome. There's a genome of a lungfish that was recently sequenced that was 93 billion base pairs in size, 93 billion. Oh, that's 30 times your size. Oh, sorry. Oh, yeah, this one's only 20 times your size. Yeah, I mean, just a mere 20 times the size of your genome. Every single one of its cells has 20 times more DNA than your cells to have it in. But now look at the redback salamander, a paltry 29.3 billion base pairs. All right, that's nothing. It's only 7 times the size of your genome. Okay. And as I say in class all the time, I mean, its size is not what it's all about. All right. It's about how you use your genome, and how many genes you have is a little more important than the total size of your genome. But you might be asking, all right, so what constitutes the difference between these two? I mean, one's much larger, twice the size of the other genome. The doubling of the size is not due. Some of you might be wondering, and be like, oh, maybe this is just a polyploid. All right. They've doubled their genome. And so they have like 4 copies of all their genes, or 2 copies of everything instead of the 1. No, this is still, they're both, they both have the same numbers of genes. It's just that one of them has a massive amount of repetitive DNA. Transposons, jumpable genes. I mean, these things that copy themselves in the genome and proliferate in the genome, making the genome explode in size, adding 30 billion base pairs to its genome. And yet you can see that having 30 extra billion base pairs doesn't really make a huge difference. Honestly, I'm pretty sure you could cut out the 30 billion base pairs out of the one over here, or this one over here. You could probably cut 30 billion base pairs out of that and end up with the same organism, and it would have the same differences between these two, because the differences between the two are probably in the protein coding genes and in a few regulatory genes. All right. Well, I should have said, sorry, any of my students watching this would be upset at what I just said. In the regulatory elements, not the regulatory genes, right? The regulatory elements are portions of DNA sequence where proteins bind to them to affect how the genes are turned on and off. All right. So, now let's go back and think about if I were to ask a young Earth creationist, hey, here's two genomes. Tell me how similar they are to each other. Calculate that. And you go like, well, let's just line up the sequences and do a comparison between the two. All right, we line them up. I got 67 billion base pairs and I have 29 billion base pairs. I got an extra 30 billion base pairs in one that is not in the other. I could just like call all of those differences, right? You know, it's like every single one of those 30 billion base pairs is a difference. You don't have it, but I have it, right? And for 30 billion times. And so I'm going to count that as 30 billion differences. So here they share 30 billion base pairs, but they don't share another 30 billion base pairs. So what's the similarity between the two? 50%. These two species are only 50% similar to one another. And this is how many young Earth creationists had their simplistic way of determining similarity, right? That whole thing about how humans and chimps are 15% different from each other, that comes from counting up every single like segment that's not found in one, but found in the other. That missing information, they call that missing data for one organism that's not found there. They count every single base pair as a difference, right? So in that case, these two organisms are 50% different. And yet, I don't think there's a single young Earth creationist right now that is serious about trying to understand species and where species come from, how they were formed, how they were created, that would say that these two species aren't actually the same organism. They're the same kind. They come from the same ancestor that was on the ark. It got off the ark and then it diverged. Well, it had to travel. Somehow the salamander traveled all the way over to North America. And then they became two different species. And so at one point, these two were the same species, along with lots of other salamanders in this particular group of salamanders. All right. There's this particular salamander kind, which contains, I don't know how, I didn't look it up, but I'm sure probably maybe several dozen species that are all very similar to one another. And so here you have two in North America that differ by 30 billion base pairs. And yet in the past, they shared the same genome. And so that many differences have occurred and they've become 50% similar to one another now. Maybe it's better to say 50% dissimilar to each other. That's pretty incredible. I'm pretty sure that if you took out and looked at all the genes, these two, I have no doubt, I haven't looked at these genomes, but I would bet that these genomes share 99.9% of the genes and 99.9% of their regulatory elements and microRNAs and so forth. All right. All the important stuff. 99 point something percent. Right. Now, when I say share, I mean they have the same gene. They still might have one or two differences out of like 1,000 or 2,000 base pairs in that particular gene. And that difference might be, hey, I got a slightly different coloration pattern because I have an A instead of a T. Right. And so you're creating these small differences you see between these different species. All right. But those differences are going to be found in those important genes. But they have 30 billion other extra base pairs that are making no difference between these organisms, other than I would say that Idohensis has a disadvantage. All right. It probably is a slightly slower growing salamander because it has to copy 30 billion more base pairs every single time it makes a new cell, and it has 1 billion cells. So it has to use a little extra energy to do that. Probably not an insignificant amount. All right. So it's and even of the 30 billion base pairs they both share, guess what? I would say they're probably only, 50 million base pairs, 100 million base pairs? At best, a couple 100 million base pairs. Of that, 30 billion base pairs are probably genes and regulatory elements. I would say 90 something percent of their genome, maybe 95% of their genome, probably all repetitive sequence that they could also probably get rid of and still look just like they look right there. All right. Just like you and I could get rid of, if we had the ability to chop out, you could chop out a whole lot of our genome and still be who you are, and not, you would know the difference. All right. So then that brings us back to, well, what is the difference to an organism? Is it fair to look at their entire genome and just say, this is the percent similarity of their entire genome, if their entire genome isn't really important? So that's why most genomicists would say, let's look at the parts that we know are important to a genome and then ask, how different are those important parts? That's why we focus so much on like just a few genes and a few real variants that differ between different species, because we want to figure out what are the actual differences that made a difference between two species, because there are a lot of differences that make no difference. And so just throwing out of like, you're 2% similar or 2% different, you're 1.5% different. Well, what does that mean? It only means something to someone who knows a lot about the genome and knows exactly what variations you're talking about and knows how to compare, right? Okay, so let's use that comparison to also compare this other species. And then we have like, oh, this other species is 1.7% different, whereas this one's 1.2% different. And that might actually be meaningful to somebody if they know those particular organisms. Yeah, so, back to this particular thing. So what is the 80%, 90%, 50%? I mean, the 50% banana thing is actually just it's not actually looking at the entire genome and lining it up. You really can't line up much of a banana and a human being. There is, we'd not be 50% similar, right? Large, massive chunks of those two genomes would be all kinds of sequence that is not shared between us, is unique sequence. So the 50% is actually, ironically, I think the 50%, I really should have gone back and looked this up, but I think I imagine the 50% is, if you were to ask, of all the genes that we share, because all living things do share some genes, right? What's the difference in those sequences? That's not looking at the actual DNA sequences themselves, right? So that's another level of asking about similarity. Sometimes you see Venn diagrams, like here's all the genes in this organism, here's all the genes in this organism, here's how many they share, right? 25% of them, the genes are shared. Usually it's more like 60% or 70% or 90% of the genes are shared. That's not telling you what the percent of similarity is between each individual sequence of those genes. All right, yeah, I know. We're kind of going around in circles now, but here's the bottom line. The bottom line is that most of these types of things are completely useless pieces of data if you don't have a full understanding of where the data points came from. I don't even know where the data points came from on this particular image. I certainly wouldn't take it as faith that all these were generated from the same form, the same methods. I know that's not the case because the mouse is 98% similar, right? That has to be a different methodology. And so you need to be skeptical of any of these numbers that are thrown out. And here's the thing, you always need to ask, how did you calculate that number? And then once you figure out how somebody's calculated that number, he's like, give me a basic idea. What basic method did you use to come to this, to get to this data point of 98% similar or 90% similar? And once you've decided, like, okay, now can we do an apples to apples comparison? Can we use the same methodology and compare these other two organisms? All right, going back to my salamanders. All right, once you've decided, like, let's compare all the protein coding genes that they have, and let's ask the question, of all the protein coding genes that we can identify in the two species, how many do they share? And you would say, like, you know what? They share 99.9% of the protein coding genes. And it's like, okay, that's all right. That's a number. That's a value. Now, what if we compare a salamander to a frog? All right, they're both amphibians. And say, let's take that amphibian, let's take the green tree frog, and let's sequence its genome. We'll find all the protein coding genes. We'll identify all the protein coding genes we possibly can. And then we'll ask ourselves, how many of those protein coding genes are the same genes as we find in salamanders? In other words, how many genes do they share? How many are in common? So like, you know what? There's 90% of their genes are in common. So that means 10% of the genes are missing in one of those two species, right? And you say, like, that seems kind of meaningful to me. Like, these two salamanders look almost identical. They seem to be using the same genetic information to make the same looking organism, whereas a green tree frog looks pretty different. I would expect that it would have different genetic packages, programs that these salamanders don't have, and therefore the 10% similarity, dissimilarity, feels about right. And you say, like, now let's compare all the genes that are present, protein coding genes that are present in a lizard compared to this salamander. And then you find out that, there's a 20% difference. They don't share 20% of their genes. And so that's one way you could do it. And you could keep comparing all other groups. And you get some sense of how divergent, those groups are. You could compare the actual sequences of those genes. Well, now I'm just going back, sorry, and repeating myself on the different methods, but I'm just trying to give you a different angle at how to do that. And I'm emphasizing that this is what you need to force anybody to do in conversation. Anyone who ever wants to talk to you about genetic similarity, you must pin down what they mean by genetic similarity first, before you can have a conversation and you can do any comparisons. Compare a chimp and a human and agree, what are you going to compare? Okay, here, let's whatever the type of calculation is, I don't really care. I don't really have a preferred method, like thou shalt use this particular method. We have different interests in what I'm interested in comparing. And so that's why we end up with these different methods. But just agree on one of them. Say, like, okay, I'll grant you, like, this is how we're going to compare a human genome and a chimpanzee genome. And if you agree on Tompkins' method, then you say, like, okay, well, now let's apply that same method to other pairwise comparisons. Take any two individuals in a species, two different species, two different, really what you think look like very divergent morphological species, and let's use the exact same methodology and see if the results are intuitive, right? And that would be like, hey, if chimpanzees and humans are 15% dissimilar, I would expect that you and I are less than 15%. And yes, that you would find out using almost any method you came up with that you and I would be more similar to each other than we are to a chimpanzee. But creationists want to say it's 15% between humans and chimpanzees. And then they want to talk about humans and beings being 99.9% similar to one another. But that's mixed methods. Right, the 99.9% is looking at just protein coding genes and looking at just sequences of the important proteins in our DNA, right? The coding for those proteins in our DNA. That's not looking at the whole genome in the same way that they're looking at the whole genome between chimpanzees and humans. And that's why I said you and I are like 5% different. And what if we got the Neanderthal genome and they find out they're 9 or 10% different? Then what do those values mean? I mean, actually, to me, they're relatively intuitive, but they don't say the same. They don't make the dramatic entrance or story that young Earth creationists are trying to make out of those values. Then more importantly, you need to ask, what's the difference between, do your analysis, but now compare a chimpanzee and a bonobo, which are sister species, right? And you got to find out they're, 9 or 10% different. And then do that comparison between a chimpanzee and a gorilla and say, let's do the apples to apples comparison, right? Actually, you don't have to do those analyses. Gutsa Gibbon has a whole big long video actually detailing in great detail her analysis doing that exact same thing, trying to do like apples to apples. I'm going to run the exact same analysis in the same way, analyze, collect the data, start with the same assumptions, and let's get some values. All right, that's a fair comparison. And you might disagree with I don't think that's the best way to compare organisms, but at least you've agreed on the same method. right? You can argue all day long about like what's the best way and the most what is the most meaningful way to tell the difference between organisms. See here, I don't think comparing the genome size of these organisms is a very meaningful thing, because I don't think that's what makes them different from each other. Their genome size is not what makes these two organisms the small differences that they have. So therefore, just comparing genome size, not a very important value. However, if you want to do it, let's at least be consistent. Compare the genome size and then go to another salamander, compare the genome sizes. What you're probably going to end up with is a really unintuitive result. You're going to find that there's another salamander that looks really different than these two that is 95% similar to one of these because the genome size is very similar. And you're going to look at it and you're like, that doesn't really make sense to me, right? You know, those values, those percents don't fit your intuition when you just look at the organisms. Now, looks aren't always important, right? Looks can be deceiving. There can be great differences in organisms even when they look the same. But in this case, I think you should get the impression that these two species have incredibly similar features. And if you compare to frog, you absolutely should expect the frog to have differences that go beyond skin deep. They go down to the DNA level. All right. Well, I thought this was going to be a little tiny short episode of Critiquing Creationism, and here I am almost an hour later. Hey, thanks for hanging out with me, and we'll see you on the other side. Bye-bye.